Good morning. My name is Anna.
And I'm Christina. Welcome to Science Tech News. Today's topic is CRISPR.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats of Genetic Information.
CRISPR is a powerful gene editing technology used to modify, delete, or correct disease-causing abnormalities at genetic sources.
Today we have two special guests here with us to tell us more about CRISPR.
Good morning, Anna and Christina. My name is Dr. Teddy, and this is my colleague, Dr. Tundra.
Hello. As you know from Anna and Christina, CRISPR is a gene-altering technology, but let's go into more depth about this powerful technology and its history.
CRISPR was first discovered by Francisco Mojica, a scientist at the University of Alicante in Spain.
CRISPR served as part of the bacterial immune system, defending against viruses. This theory was experimentally demonstrated in 2007 by a team of scientists led by Felipe Horvath.
This technology utilizes chemical engineering. It is an RNA-guided gene editing platform that makes use of a bacterially derived protein, Cas9, in a synthetic guide RNA to introduce a double-strand break at a specific location within the genome.
Cas9 is an enzyme that snips DNA, and CRISPR is a collection of DNA sequences that tells Cas9 exactly where to snip. This tool can quickly and efficiently tweak almost any gene in any plant or animal. As you can see in this photo shows DNA being cut out of the DNA strand.
Researchers already have used it to fix genetic diseases in animals, to combat viruses, and to sterilize mosquitoes. They have also used it to prepare pig organs for human transplants and to beef up the muscles in beagles.
This technology could potentially decrease and even eliminate the incidence of many serious genetic diseases.
While this technology seems totally awesome, it does raise some ethical concerns. CRISPR raises questions about the ethical merits and consequences of tampering with genomes. Some people believe that embryo modification is unnatural or amounts to playing God.
The biggest limitation of CRISPR is that it is not 100% efficient. According to the 2014 Science article by Doudna and Charpentier, in a study conducted in rice, gene editing occurred in nearly 50% of the cells that received the Cas9 RNA complex, whereas other analyses have shown that depending on the target, editing efficiencies can reach as high as 80% or more.
There are also off-target effects where DNA is cut at sites other than the intended target.
Thank you, Dr. Teddy and Dr. Tundra, but sadly, this is all the time we have today. So, what do you think about CRISPR?
I think CRISPR is pretty cool. I've always been interested in genetics since middle school. I believe that altering genes to decrease and eventually eliminate genetic diseases could really benefit the society. What about you?
I also think CRISPR is cool. Just the fact that you can modify or delete genetic diseases within the DNA is so interesting. Like you said, society can really benefit from this, and it's just the beginning. CRISPR is still new and improving each day. Who knows what will happen next?
So what do you all think about CRISPR?
Thank you all for tuning in to Science Tech News.
Check out this cool video by McGovern Institute for Brain Research at MIT.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with D. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The Crispr method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.